Good morning to you. Welcome back, everybody, to daily lectures from New Age Cosmetic White EdTech Healthcare Consultancy. I am Dr. Bobby, and we are on the subject of molecular biotechnology. We are on the topic of bioremediation and biomass utilization. So the subtopics on this would be the subtopics will be microbial degradation of xenobiotics, genetic engineering of biodegradative pathways, manipulation by transfer of plasmids, manipulation by gene alteration, utilization of starch and sugars, commercial production of fructose and alcohol, altering alcohol production, improving fructose production, cell age fermentation, isopropanol production, engineering yeast transcription, utilization of cellulose, lignocellulosics, components of lignocellulose, isolation of prokaryotic cellulose genes, isolation of eukaryotic cellulose genes, manipulation of cellulose genes, zymomonas, mobilus, hydrogen production, summary, and review questions. For many centuries, humans have believed that atmospheric, terrestrial, and aquatic systems were sufficient to absorb and break down wastes from population centers, industries, and farming. We know that is not true today. There are two fundamental problems. Firstly, how do we dispose of the large quantities of waste that are continually being produced? Secondly, how do we remove the toxic compounds that have been accumulating at dump sites in the soil and in water systems over the last few decades? So this particular chapter is good for those who are into soil technology and things like that. In fact, I too am a student of that. But I do it since more into environmental corrections and technology-based work. I think this topic is of great importance to all those interested in the augmentation of our environment. And second is how do we remove the toxic compounds that have been accumulating at dump sites in soil and in water systems over the last few decades. Governments have been trying to meet the challenge of environmental contamination by instituting anti-pollution regulations, but these rules often remain unenforced. Governments have also encouraged the 3 R's: reduce, reuse, and recycle. Researchers currently testing a number of technological strategies, including biotechnological schemes, to deal with large-scale waste such as lignocellulosics and toxic substances that persist in ecosystems. The term bioremediation has been introduced to describe the process of using biological agents to remove toxic waste from the environment. Biomass is the term used to describe the materials that are produced by the food and agriculture industries, example starch and lignocellulosics, that were discarded as waste in the past. Biomass is now being considered as a source material for the production of a variety of economically important products. And on to microbial degradation of xenobiotics. Now, the problem of toxic waste disposal is enormous. Worldwide production in 1985, just one chemical that's released into the environment, pentachlorophenol, was more than 50,000 tons. Incineration and chemical treatment have been used to break down many toxic chemicals, but these methods are costly and often create new environmental difficulties. With discovery in the mid-1960s of a number of soil microorganisms that are capable of degrading xenobiotic and natural or synthetic, from the Greek xenos meaning foreign, chemicals such as herbicides, pesticides, refrigerants, solvents, and other organic compounds, a notion that microbial degradation might improve an economical and effective means of disposing of toxic chemical waste gained credence. Members of the genus Pseudomonas are the most predominant group of soil microorganisms that degrade xenobiotic compounds. Biochemical assays have shown that various Pseudomonas strains can break down and, as a consequence, detoxify hundreds of different organic compounds in many cases. One strain can use any of several different related compounds as its sole carbon source. The biodegradation of complex organic molecules generally requires the concerted efforts of several different enzymes. The genes that code for the enzymes of these biodegradative pathways are sometimes located in the chromosomal DNA, although they are more often found in large, approximately 50 to 200 kilobase plasmids. In some organisms, the genes that contribute to the degradative pathway are found in both chromosomal and plasmid DNA. Degradative bacteria, in most cases, enzymatically convert xenobiotic, non-halogenated aromatic compounds to either a catechol or proto-catechate. Then, though a series of oxidative cleavage reactions, catechol and proto-catechate are processed to yield either acetyl coenzyme A, acetyl-CoA and succinate, or pyruvate and acetaldehyde compounds that are readily metabolized by almost all organisms. Halogenated aromatic compounds, which are the main components of most pesticides and herbicides, are converted to catechol, proto-catechate hydroquinones of the corresponding halogenated derivatives by the same enzymes that degrade the non-halogenated compounds. However, for the halogenated compounds, the rate of degradation is inversely related to the number of halogenated atoms that are initially present on the target compound. Dehalogenation, the removal of a halogen substituent from an organic compound, is the critical requirement for detoxification and often occurs by a non-selective dioxygenase reaction that replaces the halogen on a benzene ring with a hydroxyl group. This step may occur either during or after the biodegradation of the original halogenated compound. The genetic engineering of biodegradative pathways. Despite the ability of many naturally occurring microorganisms to degrade a number of different xenobiotic chemicals, there are limitations to the biological treatment of these waste materials. For example, 1. No single microorganisms can degrade all organic wastes. 2. High concentrations of some organic compounds can inhibit the activity or growth of degradative microorganisms. 3. Most contaminated sites contain mixtures of chemicals, and an organism that can degrade one or more of the components of the mixture may be inhibited by other components. 4. Many non-polar compounds adsorb onto particulate matter in soils and sediments and becomes less available to degradative microorganisms. And 5. Microbial biodegradation of organic compounds is often quite slow. One way to address some of these problems is to transfer by conjugation into a recipient strain plasmids that carry genes for different degradative pathways. If two resident plasmids contain homologous regions of DNA, recombination can occur and a single larger fusion plasmid with combined functions can be created. Alternatively, if two plasmids do not contain homologous regions and in addition belong to different compatibility groups, they can coexist with a single bacterium. Manipulation by transfer of plasmids. Bacterial strains with expanded degradative capabilities were first created in the 1970s by Chakraborty and co-workers. They used different plasmids to construct a bacterial strain that degraded a number of the hydrocarbon components found in petroleum. This strain has been called a superbug because of its increased metabolic capabilities. The CAM, the camphor degrading plasmid, was transferred by a conjugation into a strain carrying the OCT. Right? Just a second. That's the octane degrading plasmid. These two plasmids were incompatible and could not be maintained in the same cell. That's right. And as separate plasmids. However, when recombination occurred between the two plasmids, a resulting single plasmid was perpetuated and carried both camphor and octane degradative activities. The NaH naphthalene degrading plasmid was transferred by conjugation into a strain carrying the XYL xylene degrading plasmid. The NaH and XYL plasmids were compatible and could therefore coexist within the same host cell. Finally, the CAM or CT fusion plasmid was transferred by conjugation into a strain carrying the NaH and XYL plasmids. The final result of these manipulations was the generation of a strain that grew better on crude oil than did any of these single plasmid strains, either alone or in combination. Although this particular multiple degradative strain has not been used to clean up oil spills, it has displayed a critical role in the development of the biotechnology industry. The inventor of this superbug was granted a US patent describing its construction and use. This was the first patent ever granted for a generally engineered microorganism, represented a watershed code decision because it implied that biotechnology companies could protect their inventions in the same way as the chemical and pharmaceutical industries had in the past. Most of the degradated bacteria that have been genetically manipulated by plasmid transfer are mesophiles, organisms that grow well only at temperatures between 20 and 40 degrees centigrade. However, rivers, lakes, and oceans that are polluted generally have temperatures that range from 0 to 20 degrees centigrade. To test whether bacteria with enhanced degradative abilities could be created for cold environments, a TOL, toluene degrading plasmid from a mesophilic Pseudomonas putida strain was transferred by conjugation into a facultative saccharophile, an organism with a low temperature optimum. The host saccharophile was able to degrade salicylate but not toluene, and to use it as a sole carbon source at temperatures as low as 0 degrees centigrade. Now, the transformed strain carried the introduced TOL plasmid, from a mesophilic Pseudomonas putida strain was transferred by conjugation into facultative saccharophile, an organism with low temperature optimum. The host saccharophile was able to degrade salicylate but not toluene, and to use it as a sole carbon source at temperatures as low as 0 degrees centigrade. The transformed strain carried the introduced TOL plasmid and its own SAL salicylate degrading plasmid, was able to use either salicylate or toluene as its sole carbon source at 0 degrees centigrade. The wild-type non-transformed saccharophilic strain was unable to grow at any temperature when toluene or toluate was the only carbon source not shown. This temperature experiment indicates the feasibility of engineering saccharophilic degradative bacteria for use in the environment. Manipulation by gene alteration. 4-ethyl benzoate, bringing together different intact plasmid-based degradative pathways, but conjugation is only one way to create bacteria with novel properties. It may also be possible to extend the degradative capability of a strain by altering the genes of an existing degradative pathway. The feasibility of this approach was examined for the toluene and xylene degrading pathway of plasmid PWWO. This plasmid encodes a metacleavage pathway involving 12 different genes and enables pseudomonads carrying the plasmid to utilize various alkyl benzoates as carbon sources. The genes in the toluene xylene pathway of PWWO are part of a single operon called the XYL operon under the control of the P promoter. Transcription from the PM promoter by RNA polymerase is positively regulated by the Xyl as gene product, which is activated by most of the initial substrates such as benzoate and 3-methylbenzoate of the pathway. Detailed biochemical and genetic analysis showed that bacteria carrying PWWO could degrade 4-ethyl benzoate, albeit slowly, to 4-ethyl catechol, which accumulated in the medium, but no further. 4-ethyl catechol prevented its own degradation by inactivating one of the most important enzymes in the biodegradative pathway, catechol 2,3-deoxygenase. The product of the xyl gene, in addition, 4-ethyl benzoate, unlike most other alkyl benzoate, does not activate the xyl S protein. Consequently, transcription of the operon from the PM promoter did not occur at any significant extent when 4-ethyl benzoate was the only substrate. Thus, there are two major problems with the naturally occurring metacleavage pathway system. One, how to overcome the inactivation of an important enzyme in the degradative process by 4-ethyl benzoate, and two, how to induce its transcription of the genes of this pathway with 4-ethyl benzoate as the inducer. To find a mutant that could solve the second problem, a tetracycline resistance gene was placed under the control of the PM promoter on one plasmid, which also carried an ampicillin resistant gene. The xylase gene was cloned into another plasmid carrying a kanamycin resistance gene. Transformants carrying both of these plasmids were selected on the basis of resistance to both ampicillin and kanamycin. The E. coli cells carrying both of these plasmids were treated with mutagen ethyl methane sulfonate and plated onto a medium containing both tetracycline and 4-ethyl benzoate. The early cells that could grow on this medium carried on altered xyl S protein, S4 in figure 14.7a, that could interact with 4-ethyl benzoate and cause the tetracycline resistance gene to be transcribed. Thus, the degradative pathway that includes this mutated xyl S gene can be induced by 4-ethyl benzoate. To address the catechol 2,3-dioxygenase inactivation problem, the mutated xyl S gene was subcloned onto a broad host range plasmid carrying a kanamycin resistance gene and introduced into P. putida cells carrying PWWO. The transformed cells were plated at a high cell density onto a minimal medium containing 4-ethyl benzoate as a sole carbon source, kanamycin to select for the presence of the plasmid, and ethyl methane sulfonate. Cells that were able to grow on this medium produce an altered form of the enzyme catechol 2,3-dioxygenase that was not inhibited by 4-ethyl catechol. Additional analysis confirmed that the catechol 2,3-dioxygenase gene was PWWO, had been mutated, and that mutant versions of both the xyl S gene and the catechol 2,3-dioxygenase gene were required for the degradation of 4-ethyl benzoate. An important aspect of this work is the fact that the two genes that were altered, that is, those encoding xyl S and catechol 2,3-dioxygenase, are the major determinants of the range of compounds that can be degraded by this pathway. The work with 4-ethyl benzoate demonstrates that by combining the recombinant DNA technology, conventional mutagenesis, and the appropriate selection protocols, novel properties can be added to a degradative pathway. Trichloroethylene. The compound trichloroethylene is widely used as a solvent and a decreasing agent, and as a result, it is one of the most common contaminants of soil and groundwater. Trichloroethylene. So this persists in the environment for years is a likely carcinogen and is regulated in the US under Safe Water Drinking Act to a maximum contaminant level of 5 parts per billion. Unfortunately, anaerobic soil bacteria can reductively dehalogenate it to produce vinyl chloride, which is an even more toxic compound. Studies showed that some of the strains of P. putida, that is Pseudomonas putida, that could degrade aromatic compounds such as toluene could also degrade trichloroethylene. Genetic studies established that the complete metacleavage degradative pathway was not necessary to completely detoxify trichloroethylene. In fact, only the enzyme toluene deoxygenase, which normally catalyzes the oxidation of toluene to cis toluene dihydroidio, was required. 4 genes are involved in the production of a functional toluene dioxygenase. These genes were isolated and expressed in E. coli under the control of the strong and inducible TAC promoter. When E. coli cells carrying these genes were induced by the addition of isopropyl beta D-thiogalactopyroxenide (IPTG) and induced over the TAC promoter, trichloroethylene was efficiently broken down to harmless compounds by the concerted enzymatic activities of the TOD proteins encoded by these 4 genes. Although the initial rates of trichloroethylene degradation were lower with E. coli than with the original P. putida strain, the E. coli cells maintained these rates for longer periods than P. putida. It has been speculated that the basis for this difference may be that E. coli membranes are not as susceptible to damage from trichloroethylene as P. putida membranes are. In a variation of this experiment, a hybrid Pseudomonas strain with elements of 2 separate degradative pathways was constructed. Bacterial strains that can degrade the compound biphenyl use the enzyme biphenyl dioxygenase. Biphenyl dioxygenase is a multi-component enzyme encoded by 4 genes, BPH A1, A2, A3, A4, where BPH A1 encodes a large subunit of terminal dioxygenase. Anion sulfur protein, BPH A2, encodes a small subunit of terminal dioxygenase. BPH A3 encodes ferredoxin, and BPH A4 encodes ferredoxin reductase. BPH A1 and BPH A2 are associated as a heterotetramer and catalyze the introduction of 2 oxygen atoms into the biphenyl ring. Ferredoxin and ferredoxin reductase act as an electron transfer system from reduced nicotinamide adenine dinucleotide NADH to reduce the terminal deoxygenase. Biphenyl deoxygenase is quite similar in both structure and function to the enzyme toluene deoxygenase. Despite the similarities of the enzymes, biphenyl utilizing Pseudomonas monads cannot grow on toluene, and toluene utilizing strains cannot grow on biphenyl. However, when the BPH-A1 gene coding for the large subunit of biphenyl dioxygenase from P. putida KF715 was replaced by homologous recombination with the TOD-C1 gene coding for the large subunit of the toluene deoxygenase from P. putida F1, the resultant strain was able to degrade trichloroethylene. In fact, the engineered strain grew well on a range of aromatic compounds and also was very efficient at degrading trichloroethylene, demonstrating that it is possible to rationally engineer bacterial strains that can degrade a number of different compounds. The creation of degraded bacteria with novel biological activities was achieved by slightly different means, by the creation of chimeric versions of the BPH-A1 gene. In this case, one BPH-A1 gene was from a strain of Pseudomonas pseudoalkalogens with the ability to degrade only a narrow range of polychlorinated biphenyls PCBs, while the other was from Burkholderia cepacia, which can degrade a very wide variety range PCBs. Some of the hybrid genes encoded an enzyme with a wider degradation ability than either of the original enzymes. The native BPH-A1 gene in P pseudoalkali genes was replaced with hybrid genes by homologous recombination. It now remains to be seen whether these engineered bacterial strains degrade a range of PCBs on a large scale. Cell surface expressed enzymes. Currently, detoxification of organophosphate pesticides in the environment is performed by chemical treatment, incineration, or burial in landfill sites. Each of these approaches has serious environmental drawbacks. It would therefore be advantageous. Bacteria that are able to degrade these compounds could be utilized in place of the methods that are currently used. Several soil bacteria, including Pseudomonas diminuta, Mg, and Flavobacterium spp, possess an enzyme, organophosphorus hydrolase, that catalyzes the hydrolysis of many of these pesticides, including methyl and ethyl parathion, paraoxone, chlorpyrifos, diazban, coumaphos, cyanophos, and diazinon to environmentally innocuous compounds. Unfortunately, these bacteria, as well as E. coli engineered to express organophosphorus hydrolase, degrade these pesticides relatively slowly because of the low rate of uptake into the bacterial cells. Thus, a novel approach was developed to solve this problem. E. coli cells were engineered to express organophosphorus hydrolase as part of a fusion product protein that contained the E. coli lipoprotein signal peptide and the N-terminal portion of the lipoproteins and outer membrane protein A. When this fusion protein was synthesized, it was localized on the outer surface of the bacterium. These eliminated the problem of a low rate of pesticide uptake in the engineered bacterium. Cells with organophosphorus hydrolase on the outer surfaces had approximately 7 times higher activity than cells that express a similar compound of the enzymes intracellularly. Moreover, the enzyme activity of cells with organophosphorus hydrolase on their surfaces was extremely stable. Nearly 100% of the activity was retained after 1 month at 37 degrees centigrade. This concept is quite promising. However, to date, it's been tested only on a laboratory scale. Radioactive environments. "26 countries worldwide that generate electricity from nuclear power plants as of now, or should be higher now, as well as those countries that have nuclear weapon programs, have generated thousands of radioactive wastes at sites. In the United States alone, there are more than 3,000 of these sites, and it's been estimated that using currently available technology, the cleanup will require around $200 billion and take approximately 70 years. In addition to radioactivity, these sites also often have both organic and metal pollutants. While biodegradation of the organic pollutants is a logical first step in this remediation, most microorganisms are highly sensitive to the damaging effects of the radiation. Fortunately, the non-pathogenic soil bacterium Deinococcus radiodurans is naturally resistant to quite high levels of ionizing radiation. This resistance has been attributed to DNA repair process that are exceptionally effective at repairing DNA damage. Moreover, any DNA that is introduced into D-radiodurans, either as part of a plasmid or inserted into the chromosome, is also protected against high levels of potential damaging radiation. Since this bacterium can express foreign genes while growing in the presence of continuous radiation, it would appear to be an ideal candidate for the expression of bioremediating proteins in toxic environments that contain radioactive contaminants. As a first step towards developing a system to remediate organic pollutants that are present in radioactive environments, the 4 genes that together code for toluene dioxygenase were placed in a plasmid under the control of a constitutive D-radiodurans promoter. And the entire plasmid was then inserted into the chromosome D-radiodurans by homologous recombination, a single crossover between the chromosomal DNA and a chromosomal DNA fragment of the plasmid adjacent to the toluene dioxygenase genes. The integrated toluene dioxygenase was active and conferred upon D-radiodurans the ability to degrade toluene, chlorobenzene, and 3,4-dichloro-1-butene irrespective of the presence or absence of high levels of ionizing radiation. The successful expression of toluene dioxygenase, an enzyme with several protein components and including metal and organic cofactors, suggests that many less complex biodegradative enzymes systems could also be expressed in D-radiodurans, and this is tolerant of high levels of toluene and a number of other organic compounds. Once this bacterium has been genetically engineered to express the appropriate biodegradation pathway, it should be able to degrade a variety of organic pollutants in a radioactive environment. However, it remains to be seen how these engineered organisms behave under field conditions. Nitroaromatics. For many years, a large number of different nitroaromatic compounds have been used industrially as dyes, plasticizers, explosives, solvents, and pesticides. Many of these compounds are recalcitrant to break down, persist in the environment, and are now considered to be toxic and sometimes carcinogenic pollutants. For example, the compound 4-nitrophenol is formed by the hydrolysis of the insecticide parathion and is considered to be a priority environmental pollutant, in part because it leads to numerous human health problems. Similarly, the compound 3,4-methyl-4-nitrophenol is a toxic breakdown product of the agricultural insecticide phenyltrethion. The bacterium Burkholderia sp strain DNT facilitates the breakdown and detoxification of 2,4-dinitrotoluene. First, the enzyme 2,4-dinitrotoluene deoxygenase removes one nitro group to form 4-methyl-5-nitrocatechol, which is then converted to 2-hydroxy-5-methylquinone by the enzyme 4-methyl-5-nitrocatechol monooxygenase. And fortunately, the 4-methyl-5-nitrocatechol monooxygenase has a very narrow substrate range, which includes 4-nitrocatechol as well as 4-methyl-5-nitrocatechol. However, this enzyme cannot efficiently use either of the seemingly similar substrates for 4-nitrophenol or 3-methyl-4-nitrophenol. In an effort to expand the substrate range of the enzyme, the Burkholderia species strain DNT 4-methyl-5-nitrocatechol monooxygenase gene was isolated and subjected to error-prone PCR, and the randomly mutated genes were cloned into a plasmid vector. The library of mutated 4-methyl-5-nitrocatechol monooxygenase gene was transferred into E. coli cells by electroporation, and the transformants were screened for activity on agar plates containing 4-nitrophenol. Cells that contained the wild-type gene turned the colonies light brown, while one transformant of 3000 tested turned the colony dark brown, indicating that the 4-nitrophenol was being broken down. When the DNA sequence of the 4-methyl-5-nitrocatechol monooxygenase gene from the transformant that turned the medium dark brown was determined, 2 amino acids within the encoded protein had been altered. Amino acid 22 was changed from methionine to leucine, and amino acid 380 was changed from leucine to isoleucine. These 2 amino acid alterations resulted in the altered enzyme having 10 times greater activity toward 4-nitrophenol and 4 times greater activity towards 3-methyl-4-nitrophenol than the native form of the enzyme. In addition, the modified enzyme had about 50% more activity than the native enzyme towards 4-nitrocatechol and 4-methyl-5-nitrocatechol. The changes in activity that were found following the mutagenesis of the 4-methyl-5-nitrocatechol monooxygenase gene are an important first step in developing a bacterium that can effectively degrade 4-nitrophenol and 3-methyl-4-nitrophenol. The utilization of starch and sugars. Starch, the major food reserve polysaccharide in plants, consists of a mixture of linear homopolymers, amylose, and branched homopolymers, amylopectin, of D-glucose. Amylose is made up of linear chains of 1 by 10 raised to 2 into 4 by 10 raised to 5 D-glucose residues linked by alpha-1 4 bonds. The amylopectin consists of short linear chains of approximately 17 to 23 glucose units that are linked by alpha-1 4 bonds and joined by 1,6 linkages and some 1,3 linkages to form a highly branched structure that contains 1 by 10 raised to 4 to 4 by 10 raised to 7 glucose residues. The degree of branching and the ratio of amylase to amylopectin vary with the source and the age of the starch. Now we're on to the commercial production of fructose and alcohol. The major use of starch is in the food and brewing industry, where it is usually hydrolyzed to low molecular weight components before being converted into other compounds. especially fructose and alcohol, the most important enzymes for breaking down and transforming starch are alpha amylase, glucoamylase, and glucose isomerase. Together, these three enzymes account for approximately 30% of the cost of all enzymes currently used for industrial processes. Fructose and alcohol are produced commercially from starch by multi-step processes that include both enzymatic and non-enzymatic reactions as follows. The procedure begins with the gelatinization of milled grain, often corn, maize, which is approximately 40% starch. This treatment, steam cooking under pressure, exposes the surface area of the starch, thereby making it more readily available for subsequent enzymatic hydrolysis. The product of this process has a gel-like consistency. The gelatinized starch is cooled to 50 to 60 degrees centigrade and alpha amylase is added. In this liquefaction step, the gel-like starch is enzymatically digested by the hydrolysis of the available alpha-1,4 linkages to form low molecular weight polysaccharides. A high temperature is used because enzyme hydrolysis is more rapid at high temperature, and the enzyme does not efficiently penetrate the gelatinized starch at lower temperatures. The final step for the release of glucose includes the addition of glucoamylase and results in the saccharification. Complete hydrolysis of the remaining polysaccharides, including both linear and cross-linked molecules. The end product of these treatments is glucose, which may be either converted into alcohol as a result of fermentation by yeast cells or transformed into fructose by the enzyme glucose isomerase. The success of the latter enzymatic conversion has led to the replacement of sucrose by fructose, which is much cheaper than sucrose as a sweetener for prepared foods and beverages in North America. The source of starch for commercial fructose production is generally corn, and the final product is called high fructose corn syrup or high fructose syrup, although it contains a mixture of fructose and glucose. Excuse me. Although it contains a mixture of fructose and glucose, high fructose corn syrup is typically either 90% fructose for baked goods, 55% fructose in soft drinks, or 42% fructose in sports drinks, with the remainder being glucose. The enzyme alpha amylase randomly hydrolyzes alpha-1,4 linkages in both amylose and amylopectin chains, yielding a mixture of glucose, maltose 2 glucose molecules joined by an alpha-1,4 linkage, maltotriose 3 glucose molecules joined by alpha-1,4 linkages, and a series of alpha-limit dextrins, which are the portions of the amylopectin chains that contain cross-links. Although alpha amylase can be isolated from a variety of microorganisms, it is commonly obtained from the bacillus amyloliquefaciens for industrial purposes. For some applications, the enzyme beta amylase is used in addition to or in place of alpha amylase to digest starch by hydrolyzing alternate alpha-1,4 linkages from the ends of amylose and amylopectin chains. Beta amylase cleavage yields primarily maltose residues and various beta-limit dextrins. Next, on to a milestone note. Microorganisms having multiple compatible degradative energy generating plasmids and preparation thereof. Courtesy A.M. Chakrabarty and U.S. Patent 4259444 in 1981. Thank you for the note. Before the advent of recombinant DNA technology, one of the ways that DNA could be moved from one microorganisms to another was by conjugation, in which entire plasmids were transferred between organisms. Chakrabarty transferred degradative plasmids encoding all of the enzymes involved in the biodegradation of a particular compound from one bacterium to another until he had constructed bacterial strains that contained several degradative plasmids. Each degradative pathway encoded by the gene on a plasmid degraded a different organic molecule. Chakrabarty and his co-workers constructed a single bacterium that contained the pathways to degrade camphor, octane, salicylate, and naphthalene. While this work was scientifically interesting and innovative and of itself, especially since it was conducted in the early 1970s before the development of most of the techniques of recombinant DNA that are now taken for granted. The key issue for the biotechnology industry was that this invention was awarded a US patent in 1981, nearly 9 years after the application was first filed. Following the landmark decision by the US Supreme Court in Chakrabarty's favor, it was ruled that genetically engineered microorganisms were inventions that could be patented in the same way as any other invention. As much as any landmark scientific experiment, this court decision and the subsequent award of the patent has become a cornerstone of the development of biotechnology industry. The enzyme glucose amylase hydrolyzes alpha-1,3 or alpha-1,4 and alpha-1,6 linkages. However, because it is less efficient than alpha-amylase in cleaving, alpha-1,4 linkage is usually used in conjunction with alpha-amylase. The major role of glucoamylase is digestion of the cross-links of amylopectin, which results in its complete breakdown to glucose. Glucoamylase and other enzymes are used to reduce the carbohydrate limit dextrin content of normal beers to produce the so-called light and dry varieties. Although glucoamylase digestion is usually performed prior to the onset of the fermentation, the two steps may be combined. A number of organisms produce glucoamylase, but for industrial purposes it is usually obtained from the fungus Aspergillus niger, altering alcohol production. The enzymes that are used in the production of alcohol or fructose from mill grain are major components of the overall cost of the process. These enzymes are often used only once and then discarded. Thus, innovative approaches to the inexpensive large-scale production of the enzymes could lower the cost of alcohol or fructose production. There are several ways to achieve this end. Each of the enzymes could be overproduced in a fast-growing recombinant microorganism that utilizes an inexpensive substrate, thereby lowering the cost compared with production from native organisms. Variants of alpha-amylase, either naturally occurring or genetically manipulated, that function efficiently at 80 to 90 degrees centigrade could be used to allow the liquefaction step to be performed at this temperature. Heat-resistant alpha-amylase would speed the hydrolysis of gelatinized starch while decreasing the amount of energy that is required to cool the gelatinized starch at a temperature suitable for starch hydrolysis. The alpha-amylase and glucoamylase genes could be altered so that each enzyme would have the same temperature and pH optimum, thereby enabling the liquefaction saccharification steps to be performed under the same conditions. An enzyme that could efficiently degrade raw starch could be found or engineered, obviating the need for the gelatinization step and thereby saving a large amount of energy. A fermentation organism that can synthesize and secrete glucoamylase could be developed, eliminating the need to purify and add this enzyme during a fermentation. A considerable amount of research has been initiated to determine whether these possibilities are feasible. Genes that code for alpha-amylase have been isolated from a number of organisms, including beta-amylooligopharations and the high-temperature tolerant bacterium Bacillus styrothermophilus. Briefly, chromosomal DNA was isolated, partially digested with the restriction enzyme SAU3A1, and then ligated to BAMH1, digest BUB110 DNA. This plasmid has a unique BAMH1 site and carries a kanamycin resistance gene. SAU3AI and then ligated to BAMH1 digested BUB form DNA. This plasmid has a unique BAM and carries a kanamycin resistance. The cloned bank was transformed into Bacillus subtilis, does not have alpha amylase activity, and transformants were selected for resistance to kanamycin. All transformants were tested for the production and secretion of alpha amylase as follows. After the transformation had occurred, had formed colonies at 65 degrees centigrade on solid medium containing starch. The plates were exposed to iodine vapor. The colonies producing alpha amylase were surrounded by distinct halo or clear zone, indicating that the starch in the immediate vicinity of these cells have been hydrolyzed. A positive starch iodine test signifies that the transformed cell contains an alpha amylase that is transcribed from its own promoter because the vector does not carry a promoter. Also, a secretion signal is present because the substrate is too large to enter the cell. Therefore, the halo must be due to the activity of a secreted alpha amylase. The availability of alpha amylase genes from various sources will enable researchers to carry out specific gene modifications or genetic modifications that suit the needs of specific industrial process. Within the aim of bypassing the saccharification step during the production of alcohol from starch, researchers isolated a full-length glucoamylase complementary DNA, cDNA from the fungus Aspergillus albumari, and cloned it into Saccharomyces cerevisiae plasmid under the control of the promoter and transcription terminator regulatory signals from the yeast enolase ENO1 gene. A laboratory strain of S. cerevisiae that was transformed with its plasmid carrying the glucoamylase gDNA was able to express its activity and to ferment soluble starch to alcohol, thus demonstrating that the approach is feasible. Unfortunately, this laboratory strain of S. cerevisiae has a number of properties that make it ill-suited for use in commercial process, including an inability to tolerate high levels of alcohol inhibition expression of the glucoamylase DNA, cDNA, a loss of plasmids unless special conditions, selective pressure, are used for their maintenance. These problems, however, are not insurmountable. First, the level of glucoamylase expression was increased approximately fivefold by deleting a 175 base pair BP negative plasmid in a negative regulatory region from the NO1 promoter on the plasmid. Second, the plasmid was modified by deleting its yeast origin of replication and adding a segment of DNA that is homologous to a yeast chromosomal site to develop a bacterium that might be effective in silage fermentation. An alpha amylase gene from a strain of lactobacillus amylovirus that does not support silage fermentation was spliced into L. plantarum gene for conjugated acid bile hydrolase CBH and integrated into the chromosomal DNA of a strain of L. plantarum. As a result of these modifications, investigators created two novel yeast strains that performed better than a natural occurring amylolytic starch hydrolyzing yeast, Saccharomyces diastaticus, which is closely related to S. cerevisiae and can hydrolyze and ferment soluble starch. The performance of the brewer strain, which contained the integrated glucoamylase gene, was superior to that of the lab strain with the same gene on the multi-copy plasmid. This difference probably reflected the plasmid's inability with the concomitant use of introduced glucoamylase gene. Neither the laboratory strain nor the brewer strain of S. cerevisiae was able to use soluble starch unless the strain was transformed with the cloned glucoamylase gene. Both the plasmid and integrated forms, the A. avomoric glucoamylase cDNA was under the control of regulatory signals, FENO1, from which the 175 BP negative regulatory region had been removed. The plasmid was maintained under selective pressure. In an effort to produce large amounts of glucoamylase, several copies of the glucoamylase gene was integrated with the chromosomal DNA of the fungus A. niger. Surprisingly, there was no correlation between the number of copies of the glucoamylase gene and the chromosomal DNA and the amount of measured enzyme activity On the other hand, the level of enzyme activity was strongly dependent upon the sites where the genes were inserted. Thus, merely increasing the gene copy number is not sufficient to produce a large number of active enzymes. While most strains of the yeast S. cerevisiae encode only intracellular glucoamylase, they generally express only during sporulation. A few strains encode a secreted glucoamylase. Unfortunately, none of these enzymes contains a starch-binding domain. Thus, while S. cerevisiae glucoamylases can hydrolyze soluble starch dextrins, they are unable to break down the larger molecules of insoluble starch. In an attempt to construct a strain of S. cerevisiae that could hydrolyze insoluble starch and therefore be of use in a variety of industrial processes, the coding region of a secreted S. cerevisiae glucoamylase gene was fused to DNA fragment encoding a starch-binding domain from the fungus A. niger. When this construct was expressed in S. cerevisiae, the presence of the starch-binding domain increased the ability of the enzyme to degrade insoluble starch by about sixfold. While this system is far from optimized, this simple genetic manipulation is an important initial step in the development of more efficient industrial systems for the production of alcohol from starch. Low ethanol wines. In recent years, many wine drinkers have expressed a strong preference for wines that contain only low levels of alcohol. In addition, consumer preferences have also moved to wines with a high flavor intensity that are prepared from fully matured grapes. However, the juice that is obtained from fully matured grapes generally contains a very high sugar concentration, which in turn produces wines with high levels of alcohol. A number of attempts have been made to engineer brewer's yeast S. cerevisiae to reduce the ethanol content of the wine that is produced. Although a number of these approaches have successfully lowered the ethanol concentration, they typically cause the accumulation of undesirable side products. Nevertheless, one novel strategy seems to have avoided many of the problems of past efforts. By expressing in S. cerevisiae a gene NOx-E from the bacterium Lactococcus lactis that encodes an H2O NADH oxidase, it was possible to significantly alter some of the metabolic fluxes within the yeast cell. Transformed yeast cells that carried this gene within their chromosomal DNA had an intracellular NADH content that was about 75 to 80% lower than that of the native yeast strain, and oxidized nicotinamide adenine dinucleotide NAD+ concentration that was 32 to 45 degree higher. At the same time, the reduced nicotinamide adenine dinucleotide phosphate NADPH and oxidized nicotinamide adenine dinucleotide phosphate NADP+ ratios of the two yeast streams were identical. The transformed yeast showed a 15% decrease in the amount of ethanol produced. This lecture will be over in a minute or two from now, and about 2 minutes from now, and will be continued in the next lecture. The same subject of bioremediation will be continued from the completed portion. The transformed yeast showed a 15% decrease in the amount of ethanol produced, and unfortunately increased of approximately threefold in the amounts of acetaldehyde and acetic acid that were produced, which impair both growth and fermentation, as well as imparting unacceptable flavors to the wine. To overcome these side effects, researchers undertook a systematic study of the growth of these modified yeast strain under a wide range of conditions. It was observed that if oxygen was supplied to the growing cells only during stationary phase, there was a 7% reduction in the ethanol yield compared to the native strain without the inhibitory levels of acetaldehyde and acetone being formed. Researchers are now investigating whether the wine that is produced using the above-mentioned yeast strain is suitable for human consumption. Other topics to be covered will be improving fructose production and silage fermentation, right? So improving fructose production, just a general info where we cover up, finish up. The enzyme glucose isomerase should be really called xylose or glucose isomerase because it primarily catalyzes the conversion of the 5-carbon sugar D-xylose to D-xylulose, with the conversion of D-glucose to D-fructose being a secondary side reaction. Kinetically, xylose glucose isomerase has a lower catalytic rate constant and a higher Km binding constant for glucose than for xylose, which means that xylose is converted more rapidly to xylulose than glucose is converted to fructose. So that's it for this lecture, and please make notes, do adequate research to update your knowledge on this, and we'll be back with more details in the next lecture. Have a great day. Thank you. Bye-bye.